Autism often goes long undiagnosed, and when finally diagnosed, a treatment is still needed. Dr. Phil Schwartz of Children's Hospital of Orange County explains how autism is currently being diagnosed and how new research may lead to diagnosing the disorder with a simple blood test.
Autism is a brain disease, and if we really want to understand the disease and do something about it, what we have to study is the brain. And that's very difficult to do. You can't really biopsy the brain. You can do certain things like MRIs and imaging and things like that, but you can't actually sample the brain and see what cells are going wrong, etc. So we've been actually stymied for a long time of how to study this disease, where it needs to be studied. And we've recently shown, we, meaning the scientific community, has shown that you can take simple skin cells, take a little sample of skin and grow it up in the laboratory, grow up a big population of cells, and then through some Nobel Prize winning genetic manipulation, change the minds, if you will, of those skin cells and turn them into brain cells. So from any given patient then, we can have a culture of essentially their brain cells in the dish that we can study in the laboratory without ever touching their brain. And what we're doing with this then is we've sampled a whole lot of autism patients and autism with different genetic backgrounds, such as autism patients with Fragile X disease, etc. And we are growing up and making these brain cells, different types of brain cells, and studying their genetics. Now, one of the things that's perhaps not well understood is that every cell in the body has the same set of genes, but every cell in the body does not express the same set of genes. So, for example, a skin cell expresses certain genes that make it a skin cell, and a brain cell expresses certain genes that make it a brain cell. And so now we have these large populations of cells taken from rather large populations of patients, where we can query all the genes that are being expressed in a given cell type, the neuron being the functional unit of the brain cell, and then with that try to find targets for therapy and mechanisms for disease of autism. And so this turns out to be a complex computational problem in the end. What we end up with so much data that it's no longer biology anymore, it's bioinformatics.
Dr. Schwartz told us what he hopes to learn from the research.
We want a target for therapy, one where we can say, here is a pathway in the brain that's going wrong. Let's see what kind of drugs we can design, or drugs that are actually on the shelf that we can use to interact with this pathway, and try some clinical trials in that respect. The other one is a diagnostic, and that's equally important because the disease right now is diagnosed on a behavioral ground. There is no blood test for it. And what we'd really like is a chemical lab test that we can do from blood, something simple, where we can say yes, they do or they do not have autism. Then there's one more thing, and that is that we know that autism isn't a single disease. It's a spectrum of diseases. So it's actually better called autisms than autism. And so we can predict, we're not sure, but we can predict that different autism patients will react to different drugs different ways. And by doing this genetic analysis, what we're going to try to do is something called pharmacogenetics, which just means drugs as applied to genetics. And before we even start therapy for a given child, analyze his genetics and pick the right drug for that child. So patient-specific therapy.
Dr. Schwartz is hopeful from the progress currently being made.
There are an awful lot of people working on this, and we are homing in on those parts of the brain that are being affected. And we're developing really good tools to query those parts of the brain. So I'm very hopeful that we'll see some great progress here, not in 10 years, but I think in 2 to 5 years.